Okay, I think this is all very impromptu, by the way, and I'm not even exactly sure how this introduction will come out, except to start by saying I've known Tony for probably at least 30 years. And I'd also want to begin by adding that a lot of what I've been able to accomplish in the Sedge world is really thanks to Tony. He's been the front man, gathering innumerable interesting specimens and sending information my way, and providing me with hospitality in Ann Arbor. I remember with great fondness having evening dinners with his wife, Susan, and Tony, and she loved to put on a nice spread, very special times. And Tony, like myself, has lost his wife in this last year, so these are things we will carry with us. Tony is the consummate botanist. Bill McKnight was actually overwhelmed yesterday when we were out in the field, and Tony actually was putting sedge portions up to his tongue and feeling them, and trying to understand what the textures were, and it just I say Bill McKnight just went crazy over this. One of my most fond recollections of being in the field with Tony was when we were doing research on Carex missouriensis. And we'd had a long day in the field in Illinois, and we came to what was going to be our last spot for the day, and suddenly the tire was going flat on his car. Tony is so enthusiastic in the field that he'd say, let's find a stick and we'll stick it in the hole, put it in the hole in the tire so it doesn't go flat. And meanwhile, he's starting to charge around and look at what the sedges are in the area. And you know who got to change the tire? His wife, Susan. So just to get that sense of that, that enthusiasm, that deep in wanting to find everything that's to be found out about a particular habitat, a particular site, and particular plants. That's the Tony Reznicek. So I'm really very pleased when he contributed a chapter to the book. Without his work, the book would not be what it is, as well as Andrew Hipp, which I'll try to embarrass tomorrow. They made the book very, very special. And so it's with great pleasure and great honor that I get to introduce to you Tony Reznicek.
Thanks, Paul. Those were there were a few things I was actually trying to blot out of my memory, but yeah, I'm going to talk about evolution in Carex, and I actually had some difficulty with conceptualizing this talk. Bill is a hard taskmaster, so he said, well, you shouldn't be just going on about what's already in the essay. People could read that. And so I ended up kind of reworking the talk a little bit, I think, to give it a broader framework. I see we're a little late. That's what the donut hole is for. Every one of these speeds up the talk by about 3.5 minutes. So we're good to go. Oh, can everyone hear me all right? All right. I usually wander about. Let me get this adjusted here. All right. So, I'm emeritus at the University of Michigan. I think that I'm going to change my nameplate to read archaic and unnecessary instead of emeritus. But hopefully, one thing that I can do is give you some framework, maybe extend a little bit some of the commentary that Bob Molenbrock had about morphology, and of course, basically look at sedge evolution. Now, I purposely picked this picture because, it shows how sedge evolution is ongoing until relatively recently. This plant, Carex fraserianus, was in the genus Cymophilus. So rapid evolution here, maybe less of our knowledge, less of sedges than our knowledge about them. So, and that's what I wanted to bring out. And I'm going to give you some background information on how we study evolution to start with. Is that working? There. It starts with the naming of plants. And the naming of plants starts with this guy, Linnaeus. And he had many innovations in terms of botany and zoology. But I think one of the most important, the reason that we now name plants the way we do is the information retrieval system of binomial nomenclature. So we have genus and we have species. There they are. You have to slow down on the road a little bit to catch those, the differences. We'll see both of them growing together today. So, so this is how we see and find out and retrieve information about the natural world. Now, people may think that phylogenetic trees in a broad sense, perhaps, I'll generalize, are a new innovation, but this is actually conceptually not true, because here is how the binomial plant classification actually works. And you will see that it is hierarchical, which means nested. And you will also note, if you pulled it upright and filled in the branches, that it's a tree. And so trees conceptually have been part of biology from the beginning, and ours are now phylogenetic trees, but this is not a new way of looking at biological information. But the problem is the correlation and the linkage of the classification tree with a phylogenetic tree, because the plant name is linked to this hierarchy. It encapsulates it in the sense that the genus and species encapsulate the upper level groups that it's connected to. And this, of course, is kind of a weakness. Why is it a weakness? Because that hierarchical linkage means that if our understanding of the classification changes, in many circumstances, we're obligated by the structure of the Linnaean system to also change the name. So here is where the kernel of the problem comes in terms of the nomenclature of plants. And of course, this is a big concern for everyone because changing names are annoying, they're confusing. Often it takes 3 cups of coffee to get past a new classification. So I wanted to not only give you an idea of why these changes happen, but also a little bit about why they are necessary. And Bill also warned me not to use too many big words like synapomorphy and paraphyletic. So this is not going to happen. But I am going to define a few things about classifications. But before I get into that, I wanted to point out a couple of other very important things. And they have to do with morphology in a very indirect sense, all this hierarchical classification that we have used, for example, in all those wonderful books that we learned in Gray's Manual of Botany, Gleason and Cronquist's Manual, all that had its roots, its origins, before Charles Darwin, before we understood evolution and the basis of inherited variation. It was based on correlating and describing patterns and discontinuities in the variation we observed. And it worked astonishingly well. Part of the reason it worked very well, I think, is because human beings are astoundingly good at seeing patterns and seeing subtleties. This is doubtless related to our origins on the African savannas, where understanding and seeing patterns and learning them was really crucial when human beings were actually a part of the environment in a very real and literal sense, not able to separate ourselves technologically from it. But there is a problem. And the problem is that you can easily be fooled, even with all our tremendous skills and intellectual powers, by things like convergent evolution, parallelism. Some environments are so stringent that there's only a few ways of dealing with them, like aquatic lifestyles. So aquatic plants all converge on the same look. And sometimes this convergence is quite deep, internal. Hummingbird pollination. All hummingbird flowers fundamentally look alike, and that's convergent evolution. Now, of course, we like to think we're more confident about our views of plant classification. Why? Well, modern systematics is really, in a broad sense, the path to studying evolution. And we really focus very much on a deep understanding of variation, that is morphology. And there's three things that I think have come to the forefront in all this, which aren't often appreciated. We often think everything is due to the fact that we can now dig down and look at DNA. That's only one element. The two other elements are that mathematicians who never hike and smoke too much and live in dark rooms have developed incredibly complex algorithms, mathematical techniques to look at these giant patterns of variation in chromosomes and organize probabilities of how they evolved, how they fit together. And also equally unappreciated is the fact that these techniques require incredibly complex and mathematics that requires very powerful computers to assess. So those three things are basically why we think we can do a little better. And this is what you're accessing. And there's nothing different about the characters you get from DNA, from the base pairs, than the characters you derive from morphology. What's different is that how many morphological characters can you measure on a plant? Well, if you're super enthusiastic, you might get a few hundred, and you'll need to go to an eye doctor afterwards because you'll have been staring at a microscope forever. So how many characters can you get from the genetic code? Millions, 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 millions. This is part of the reason why it's powerful and why we can't be so easily fooled when we look at classifications that link back to molecules. There you are. Computers that are primitive couldn't possibly do this, but this is basically what you're looking at when you're looking at a phylogeny. It's a hypothesis generated by these complex algorithms. So how do you go from that to a classification? Therein lies one of the difficulties. So let's have a look. Boom, boom. This is a rapidly evolving organism. There it is, it's finished. And now you have to make groups. How are you going to take this and make a group? Well, you know, you can do that. That looks cool. Very nice. This would be called monophyletic. That's one fancy term that I'll define. It means that each of the groups includes only ancestors from one lineage, only descendants of one lineage, and it includes all the descendants. Both of those are important. Why is that relevant? Why is it more relevant than, for example, looking at corolla structure? Well, think of it in terms of predictive power. If your family includes somebody who is adopted, you are immediately aware that the predictive power of looking at your parents for example, genetic conditions is not going to apply at all to an adopted child. Your family is not monophyletic. So this is why I would say things like morphology always make more sense in the context of groups that are structured like this and in the context of applying predictive power to all the features that the plants or animals or any organism has. So this is one argument I'd make. This does not mean that there's only one way to classify plants. This would also be fine. So you're always going to have to deal with people who have somewhat different views of how to break up groups out of these phylogenetic diagrams. And the branching here is basically in the same vein as the hierarchical tree structure of a classification. And what we're really doing is fitting those together as best as we can. So here we are evolving again. So what's wrong with this? Well, you can see that group ADC is actually formed of descendants from two separate lineages. This, of course, will reduce its predictive power, and we would term this polyphyletic. So we'll do one more. There's a group which all of group A, D, E, and B are in the same group, but C is separated out into another group. Well, your group does not contain all the descendants One is split off. If you think of it as a plant, it might be something that has a unique feature, like hummingbird pollination, like an aquatic habitat that has dramatically altered its morphology, and you think it's different enough that it should be separated. So, but what's the problem with doing that? Well, there's really two problems that people would point out. One is that such groups are arbitrary. In other words, there's no rhyme or reason to why you pick one or the other in a genetic sense. And the second is who gets to choose? They're subjective, and different people will choose different things. And one of the big complaints about past nomenclature systems is they're so subjective. Each taxonomist will have their own view. So this is the basic problem. Not everyone believes that paraphyletic groups which include only descendants from one lineage, but not all of them. Not everyone believes they're bad, but most people do. So I would say that's the bottom line. The genotype supersedes your phenotype. So, and I think that no one can argue that really it's your genes that determine who you are, not the color of your shirt or the length of your hair or whether you have a beard or not. So this is basically why systematists really do believe in phylogenetic classifications, their predictive power. So what do we learn from that? Well, this is what it looks like for the entire flowering plants, the big picture. And there are our favorite plants, the poales, grasses, sedges, rushes. On a worldwide scale, just quickly, there's a whole bunch of them, about 21,000 species. And these are most of the wind-pollinated monocots. So this is really all one big group. That's kind of what it looks like. The sedges and rushes are sisters to each other. Grasses have relatives with weird names. That's not part of the old poem that begins with sedges have edges. And then there's a whole bunch of other crazy stuff down there, interesting plants. So sedges are really interesting because of their highly specialized flowers, which is a huge problem for taxonomy because there's nothing to them. I mean, this is it. This is all you've got. So how are you going to build a classification on that? So this is why people who work with grasses and sedges, I think, were to some degree early adopters of phylogenetic systems because they offered a lifeline. You could climb out of the water with them and get somewhere. Now I've drawn a typical sedge flower, but that's a lie because we're talking about Carex, and Carex flowers don't look like that. There are a lot of Carex, about 2,000 species. So I don't have time to cover them all. There's several interesting things about Carex, though, that I do want to quickly point out that aren't always appreciated. We know grasses, for example. They're warm season grasses, cool season grasses, different photosynthetic pathways. All Carex are cool season plants. Almost all of them are native. Almost all of them occur in just a few habitats, which of course naturally begs the question of why are there so many? Why wouldn't 50 do or 20? So it really is an interesting point when you think about it. With all this uniformity on a large scale, they're all perennials, they're all cool season plants. They're all native, which implies a certain amount of conservatism. Why are there so many? So this is actually a problem that has plagued people thinking about sedges for a while. And the flowers are always unisexual. I mean, that's probably a good thing. It may help a little with evolution. But, and they've got this crazy structure that is unique called the perigynium. It's a fused profil, basically a bract, first little leaf on a branch, and it's tubular, and we'll see some antecedents to Carex when we get out in the field. It has 2 veins and 2 keels, which means a 2-pronged apex. And it forms this tubular structure through which the style and stigmas protrude. There's no anthers involved because, well, it'd be kind of silly to shed your anthers inside a perigynium. What good would that do you? So I want to point out one thing, and that is when first learning Carex, people are plagued by all the strange craziness that sedge inflorescences do when they're stepped on or mowed or otherwise molested. And it's because that perigynium is not a simple structure. It's a condensed, highly condensed branch. And there's a lot of complexity and a lot of things that can go wrong with development. So, what's a profil? That's the profil, and there's a branch, and there's a leaf. So, if you do a cross section of it, you'll see stem, branch, leaf, and this tube is the profil, and there are the 2 keels or veins. And this is what it looks like on a scirpus. And we'll make sure to look at one of those. There it is. That's what a Carex perigynium is, fundamentally. And, that's kind of dark. Basically, what we think has happened, one scenario is you had a branch and a profil. There's sometimes flowers in the profil, in the axils of that profil. And if you chop off this part, you end up with a tube with a branch sticking out and a flower. And from there, you can easily see how you'd get something like a Carex inflorescence structure. And if you look deep into perigynia, you actually see all the remains of that complex structure. For example, there's the ovary, there's the ovule. This is the remnant of that branch that used to extend out. And there's that bract, the profil. And there are the 2 keels. And you can see now the answer to a question that I suspect has been keeping some of you awake. Why, when Carex perigynia have teeth, there are always 2. And that's because of the profil being 2-veined, 2-keeled. And if you pull this out with very delicate dissections and magnify it further, you can see this was a remnant of a branch. And that branch used to have all sorts of stuff on it. Damaged the plant, and that can regrow and produce all sorts of crazy things off the end. So there's all sorts of things that can go wrong with a carex. So people were commenting a little bit about eating sedges and sedge-based menus. Why don't we? Well, sedge achenes tend to be extremely heavily silicified. If you wanted to chew on most of them, you would quickly wear your teeth down. And it's also conserved. That means uniform across the entire family. So that likely means that it's really deeply embedded in the genome and is not going to be easy to change. So how have carex overcome the fact that we can't eat their achenes for breakfast, unlike grasses? Well, they've done this by abandoning changing the fruit, the achene, and utilize that bract, the periginium, and that is easily modified into innumerably variable structures. So there's the periginium. Note the two teeth. There are the achenes. When people working on fossils ask me about identifying carex fossils, first thing I ask is, do you have periginia? And if they don't, I don't give them much hope because the achenes are all alike. They've just been left alone. There's very little alteration. But periginia are tremendously variable, all over the map. Trichomes, look at that fantastic two-toothed apex. Trichomes, papillae, tubercles, there's everything you can want on a sedge periginium. And there's one with lots of papillae, Carex buxbaumii, hair, nerves that are embedded. This is Carex conoidea. So all sorts of things. Carex tonsa with this whitish eliosome-like base. Carex blanda, at least on some individuals of blanda, if you go out there on a moist moonlit night, exactly when it's fully mature, they're puffed up and succulent. You press it on a herbarium specimen, all the water goes away, you can't see it. So there's just a tremendous amount going on with sedges in their periginia. And all of this diversity is going to be driven by two evolutionary drivers. One is dispersal, which carex do magnificently, and the other is protecting the ovary when it's young and soft and tender from parasites. Various kinds of flies and wasps both will parasitize sedge achenes as well as fungi. So a lot of the weird structures we see, hairs and that, are probably there to protect the periginium from that. Look at the crazy variation you get in periginium. So fantastic. And I, in the chapter that I did for Paul's book, I showed some crazy Asian sedges, thanks to some friends who've done field work in Vietnam, which is the world's center for crazy weird sedges. But it's not the only place in the world where crazy weird sedges occur. Here's a couple from Latin America, Carex holtenii. Look at this fancy complex branching. This was actually a species that was thought to be extinct until my colleague Socorro Gonzalez and I did a bit of field work and discovered that it was just being driven by. That's the story of sedges' lives. They're driven by. And this is this probably is the most intricately branched carex in the world, Carex fecunda. This is not the inflorescence. This is just one branch off the inflorescence. So multiply those branches into a big honking panicle, and it's amazing, just amazing. But life is simple in Indiana. So didn't I see that on a billboard driving down here? I thought so. I can't remember. But anyway, we basically have only two types of Carex, subgenus Vignia, subgenus Carex. Look at all those differences. And, when I do Sedge classes, my goal is for people to recognize the subgenera driving by at freeway speeds. The species sometimes require slowing down further. So there's a lot of differences, and life is easy. There's a typical Vignia, male and female flowers. The males are hidden. More Vignias, all sorts of structures everywhere. Sometimes the males are on top. Sometimes the females are on top. So, they were ahead of the game. And subgenus Carex is very different on a drive-by basis. Got lots of different inflorescence structures. And many of these, because they tend to be larger, are some of our most striking plants. Look at this Carex grayi, fantastic. Shortiana, what a weird looking thing. We'll see both of these in the field and be able to appreciate their weirdness and subtlety with no problem whatsoever. Now we come to some interesting plants, because one of the things that phylogeny has taught us is that there's a greater diversity of large groups in Carex subgenera than we previously recognized. And here's a couple of representatives from Indiana of two of the new subgenera, Carex leptalia. This is a plant that's really quite high on the cuteness quotient. This is something akin to the coefficient of conservatism, but actually applies only to sedges and small ones. And of course, Carex jamesii. We'll see at least Carex jamesii in the field. And these are what's been recognized all long ago as remnants of ancient lineages in Carex, but where they fitted in the phylogeny was completely unknown, and now we have some idea of how they fit together. And that's the key. Learning how they fit together, that's the key to understanding morphology. It's not that we've abandoned morphology to develop this new classification. We've actually reached the point where morphology, in light of our understanding of relationships, makes a lot more sense. What's interesting about these, all these unispicate species that are in their own subgenera, two of them in fact, are all androgynous. If you find a unispicate species that's unisexual or gynecandrous, it has been reduced from members of subgenus Carex or Vignia. So all of a sudden, more things make sense. It's really useless to look at phylogenies in PowerPoint because, like all good scientists, you have to have diagrams that are completely unreadable to the audience. And so phylogenies are like that. There's a nice megaphylogeny of Carex that fits together like that. But there are some interesting elements that I kind of want to pull out. Long ago, people suspected, well, by people, I guess I mean me and my friends, that's Carex people always say, people suspected this, which means all 10 people that are working on Carex. So what was Bill talking about? The community of biologists being a striking minority. Well, you know, we're really more than a striking minority. So all of us, all 10, suspected that Carex grayi and intumescens were only distantly related to the Lupulini. Nobody was willing to shift the classification because we didn't know what they were related to. So here we have some answers. That's what's great about phylogenies, answers, and answers that you can then use to make sense. What else? Oh yeah, Carex frankii. You know, that's placed with squarosa and typhina in one section. Well, where are they? They're not even on this diagram, but we know that these two are closely related. And very isolated plant, which we'll also see in the field, Carex prasina, looks like nothing else in the flora. And that's because it's related to nothing else in the flora. And here's where it's kind of distantly related to these things, which are way over in the Pacific Northwest and off into Asia. So how did it get here? Well, that's where the dispersibility of Carex comes in. It was probably, if not an ancient relic, which it probably isn't, it was probably bird dispersed long, long ago, well before the Pleistocene. Oh yeah, there's where Carex squarosa and typhina are. And guess what they're right next to? Carex shortiana, even though they don't look alike. And now we know why this bizarre hybrid, Carex demii, is plausible and expected even, because these are each other's closest relatives. And again, there's a fact that looking at the phylogenetic trees discloses suddenly morphology can be applied to this, and we can understand a lot more, and we can figure things out. Are there any extant occurrences of Carex demii in Indiana still? Does anyone know? Have to hunt for it. It's a really interesting hybrid, and it still is extant, I think, in Illinois. So that's, you know, that's really cool. And one thing that has changed, I think, and is another paradigm that has shifted in systematics, not always to the benefit of systematists, and that is to work on these huge phylogenies and huge worldwide genera like Carex really requires ramping up the level of collaboration. Systematists have generally been solitary creatures, living in tunnels in the dirt like some bees by themselves, and we can't do that to work on these phylogenies. We need to be collaborative. So here's a project where I contributed just a tiny amount. This is also a problem because these collaborative projects, you're not viewed as being particularly relevant when you put that on your CV. You are just one of a number of worker bees, so to speak, and individual author papers will count for much more. This is why it's hard to do taxonomy and get acknowledgement from university administrators because they're so numerically score oriented. Well, anyway, Carex subgenus Uncinio, once we know the groups and the subgroups, we can start zeroing in on them and start learning a lot more about them. And here, this is remarkable because this is a group that is almost exclusively Southern Hemisphere, and it's crept into North America. And in Eastern North America, there are these species. And you'll notice that Indiana is represented I mean, it's a fantastic and really interesting group, very cryptic speciation going on. You don't have this plant, very disappointing. This is the craziest looking Carex in North America. Look at those weird evergreen leaves. It's insect pollinated. It's just got all sorts of crazy things going on with it. But you do have Carex jamesii, and we'll see that, and we'll see what you can see about the morphology. We'll also see Carex timida and Wildenoi on the field trip. So, you know, you do have these sedges. I want to point out one other wonderful thing that you have in Indiana, and that's you've now got a great book for identifying sedges right over on that counter, so you have to buy it. You can get it signed. Everything is all fitting together. So it's another example of how phylogenies solve all the problems in the world. Well, maybe not all. All right, thank you. See, that donut hole did it. Got a few minutes for questions. Am I okay for a few minutes of questions? Yeah.
So what are we doing with sections now?
Oh, you had to ask that. We're still thinking.
Okay,
Yeah.
thanks.
The problem with the sections is, of course, when everything is kind of shaken up and rearranged, because nomenclature is based on types, we have to figure out what the names that apply to that section are, which are the earliest ones, and this is more complicated than it seems because there's no good indices to sectional names, unlike, for example, for species and genera. So it's kind of messy. So, but we're working on it. The big phylogeny has got a lot of information on sections. We think we have a handle on it and have it all cleared up. Well, you told me to turn off the phone and I forgot. But anyway, I have a nice.
The ray gun is right there.
The ray gun's right there. I have a nice quiet ring, so it's no big deal. Anything else? All right, I'm gonna get you back on time.